Welcome, everyone. It's my pleasure to introduce Casey Huang. I know Casey for a long time. We shared an office for like 3 years or 4 years or something. I was a grad student and Casey was a postdoc, so I learned a lot. Casey did his bachelor's at Caltech in physics, then got his another degree in Cambridge, a master's, and then a PhD at MIT, all in sort of hard condensed matter. That's the right word. And then moved to, I think, during his PhD, started moving towards biology, and has ever since sort of done really, really exciting stuff. He's gotten, NSF Career Award, NIH Innovators Award, and et cetera, et cetera. So, but it's a pleasure to have him here. To you.
Thanks, Sid. Since Sid dated both of us, or at least relatively dated both of us, I will note that Toronto must be good for your soul because there's a huge gradient in gray hair between me and Sid. So I don't know. Somehow I took the wrong path. Thanks for having me. It's been a real pleasure talking to people today. And I wanted to just give you a brief background on our lab to start with, because we work on a lot of different topics and we've kind of evolved over time. So this is a cartoon that Jorge Cham, the guy who makes PhD comics, made about our lab that was focused on this thing that fascinated me when I moved from physics into biology, which was how and why do cells, particularly bacteria, have so many different shapes and sizes? And why does it matter? And what I found real appealing about this was that we could poke and prod the system in many different ways, including through theory and computation. But we used genetics to do things like adjust the size of E. coli cells and determine what environments bigger cells are more fit in. But we also use things like mechanical perturbations through osmotic shock to suddenly change the physical environment of cells. And in doing so, discovered things about, for instance, how cell division is coupled to the mechanical environment. And really, the goal was to try and synthesize everything, just like many of you do in many systems, into a rule book about how cells grow. And I think one of the things that I really emphasized when I was talking about these topics was that it's an intrinsically multi-scale problem. You have to care about the molecules. You have to care about what the cells are doing. And so I used to show pictures like this, where we would talk about how we would integrate both molecular dynamics simulations and single molecule imaging into whole cell pictures of growth. But we'd also try to use the phenomenology of cell growth to try to infer underlying mechanisms. And where I actually started my postdoc was developing computational models of the exoskeleton of bacterial cells, the cell wall, to try to predict what sort of mechanisms were consistent with the behavior at both the single molecule and cellular scales. And I think this question is sort of how every person interested in morphogenesis ultimately couches part of their research in terms of bridging of scales. When I was at Stanford, I actually started at the same time as a good friend of mine, Justin Sonnenberg. So he's a glycobiologist, and he studies how diet affects the gut microbiome. And we taught a class together on microbial ecology. And this was actually one of the images that we used to drive a student project. So this is actually for a project trying to quantify the biogeography of the mouse gut. You can see here mouse gut epithelial cells stained in blue. It's surrounded by this thick mucus layer in green. And there's this huge, dense microbial community outside where different species are stained in different colors. And I really found this entrancing because there's so many questions that I think from a physical perspective, we'd love to be able to answer, but also from an ecological perspective, the how and why starts to come up again. And so we started to reconceptualize this multi-scale approach, thinking maybe about the parts being the different species and cells, trying to understand what's going on at the tissue and organismal level. And very quickly, we realized you also need new tools to bridge the gap. In particular, you look at that biogeography image, and you start to ask, well, what exactly am I even trying to quantify in this big image? And so this now motivates a lot of what my lab does. But I've had students come to me and say, OK, we work on so many different scales. Like, is the goal really to bridge all the way from molecules to hosts? And I think that sounds, in some sense, a little bit silly. But actually, I would also like to argue that shouldn't be necessarily off the table. And in fact, as physicists, this shouldn't even seem particularly daunting because this pales in comparison to the scales that, in this department you think about in terms of particle physics, in terms of cosmology. And the reason I really emphasize this to a biological audience is because I think there's some intrinsic uncomfortability sometimes with complexity. And what I hope to, if not convince you of, at least seed in your brain, is that systems like the gut microbiome actually represent a system where we can start to extract the most important degrees of freedom, and hence take what is a seemingly complex system and actually make predictions, quantitative predictions, by knowing a few key observables about the system. Right. So the first part I want to tell you about is work done by Po Yi. He was a former postdoc. Now he's running his own lab at Westlake, and a grad student, Taylor. And it's really about trying to predict the assembly of gut communities. And so it was motivated, again, by pictures like this. This is from Ian Devlamic's lab at Cornell. What they did was to take dental plaque and just put it down on a slide and then stain the different species different colors, right? And what I think is beautiful and entrancing, again, is to start to postulate why these cells are laid out the way they are, right? You can see, for instance, that these green cells have these orange cells innervating this small microcolony. And perhaps that's because there's some mutualism, like cross-feeding, where the green cells are providing the orange cells with something. And by contrast, maybe the green cells are staying away from these purple cells because the purple cells are making some toxin that is actively inhibiting their growth, right? And you can make a long list of all the potential interactions that these microbes could be experiencing with each other. And I want to be clear that it's almost surely the case in this system, and probably in every ecosystem, that to some degree, all of these are playing some role, right? But I think the central challenge should be, can we determine what the most important factor is, and can we quantify that in some meaningful way, right? And the thing that I would like to argue is that you absolutely cannot ignore the fact that there's a limited supply of nutrients, and they are all fighting over that limited supply. And so resource competition is sort of an inevitable part of life as a microbe in a community. So how would we go about studying this in any reasonable way? So many years ago, we had started to develop in vitro models for studying the gut microbiome. So what we did was to take germ-free mice that had been inoculated with a human fecal sample. These are called humanized mice. And then they had been treated with antibiotics at different time points, and we took their stool and we came up with a protocol for passaging these communities in a way that would be both stable and robust and mimic the microbiome of the mouse that they came from. So this is one example here where the fecal sample of the mouse is shown here on the left. And then each one of the bars after that is one passage in vitro, where the community goes from a dense community back and diluted into fresh nutrients. And what you can see is that it stabilizes very quickly. And aside from this switch of this teal species for this purple one, there's actually a very strong correlation between the abundance of each family in vitro and the abundance in the mouse. And we find that that's generally true in this specific nutrient environment. So we think this is a reasonable mimic of what might be going into the assembly of this set of microbes. We can even do one better. So we can take that undefined community. We isolated 15 species that represent about 70% of the abundance of the full community. And then we find that there are many different families. They have many different growth behaviors. But then if we put those 15 species together and we passage them until they reach a steady state, we find that the composition of that reduced reconstituted community actually mimics what the composition in the full community, right? So we think we have 15 species that mimic a more complicated community, that they themselves mimic what's going on inside mice. So that's going to be our starting point. By the way, I should say, if anyone has any questions, I know this is a broad audience, feel free to shout out at any point. I'm happy to pause. I got lots of good stopping points for my talk. So what Po Yi and Taylor did then was a really classic, simple ecology experiment to try to assess whether interactions are taking place. So they took every species and grew it by itself, and then grew every pair of species. And what you find is that if you add up how much the two species grow by themselves, and then normalize by how much the co-culture grows, right? Basically what this is saying is that if they don't talk to each other at all, then that ratio should be 1. And so if I subtract that from 1, I should get 0. And that would be a non-interacting pair, right? And what you find if you do this is that almost all interactions are negative, right? They're inhibitory in nature. The problem is you have no idea why. And a very simple reason could be because they're competing for some of the same nutrients. So what Poyi and Taylor did was to figure out how to just simplify those two. Yeah. I mean that you put the two species together in a tube with equal volumes and then just allow them to grow together. Yeah, so in this tube here, yeah, they could be producing toxins, they could be producing things that benefit each other, and they could be competing for nutrients, right? And so that's why what we find in general is that the amount that the co-culture grows is less than the amount that you'd expect from the two growing by themselves. So what Poyi and Taylor did next was just to add an arm of the experiment where every species was grown in the spent medium of each other species. So we allow species A to grow as much as it wants, and then we take that leftover and grow species B. And the idea is the following. If we think of all the nutrients in the environment as being either things only A can eat in blue, only B can eat in red, or shared nutrients, then A will eat the blue and the shared resources, and what will be left over is the red. That's if the only thing that's happening is nutrient competition, right? And so in that model, what you'd expect is that if you add these two quantities, it should be the amount that it grows in co-culture, right? So now if we calculate this quantity, which is that sum divided by the co-culture, now what we find is the vast majority of interactions are statistically indistinguishable from null. You can see the distribution here. And in fact, if we just use this data to seed a random consumer resource model, this is a model that's based only on resource competition, with the noise of our experimental measurements, you get the gray line, right? So what this is telling you is that the distribution of effects statistically looks a lot like nutrient competition is playing a dominant role. It's not the only thing that's happening. You might say, look at these positive interactions here. Those are interesting. Those are potentially important, and we'll come back to them. But it's very motivational to think, okay, maybe we've sort of like found the first principal component of interactions, and that's competition. So what do we do with that, though? So what we did next was to take every species and do untargeted metabolomics. So this is now comparing relative to the fresh media, how many things have been depleted or eaten, and how many things have been produced. The depleted things are blue columns, and the produced things are in red. So I want to note a few things. So each one of these rows is essentially a metabolic fingerprint for that species, okay? And what you can see here is that of the 18,000 features that we can detect, only a small fraction of them are being produced compared to depleted. And in fact, an even smaller fraction of them are being produced by one species and eaten by another. So it looks like there is very little cross-feeding that's taking place. But the other thing is that every species both has a sort of uniqueness to its fingerprint, but also some overlap. So for instance, these species up here are actually quite in close competition, and you can see some of the overlap just by eye, right? So what Poyi and Taylor realized is that far be it from being this additional complexity, this is actually an opportunity to use a statistical physics approach to try to simplify our picture, right? And so the idea was, can we use this data to make a quantitative prediction for how much a community should grow and what the dynamics of the community should be? And so if we think of these, in this sort of mindset, the first thing they did was the simplest, dumbest thing you can imagine, which is just to compare the amount of growth that each culture exhibited compared to the predicted growth based simply on how many blue lines there were, how many things were depleted, right? And you might say, well, no two metabolites are the same, and that's absolutely true. But fortunately for us, we got 18,000 metabolites, and the idea is that maybe all those differences just get averaged out. And the answer is not bad, right? There's definitely some dispersion. There are some things here we're predicting very poorly, but on the whole, we're doing pretty well. And so that, again, was motivational. So here's where we want to be predictive. And so what we did was to take the same picture I presented to you before with two species and just took it to its logical conclusion. So imagine you have three species, then you can have a blue resource, a red and a green, and the pairwise resources, and the ones that all three are sharing. With 15 species, there's 2 to the 15th so-called niches, I'll call them from here on out, right? And the idea is, if we can identify all these coarse-grained resources, then maybe what we should be doing is to build a very simple model based around those resource groupings. Okay? So this is a consumer resource model where we're just tracking the abundance of species X. X is growing by depleting resources Y with some rates R. And as it does so, it depletes the resources Y. Just the simplest thing you could imagine. This is what happens now if you take that metabolomic set of profiles and you just now group everything together in the most logical way. So I can take all the things that are only eaten by species 1, 2, 7, 11, and 14, and I'm going to call that one niche. Okay, and that's one of the columns along here. And then up the top is how many metabolite features are actually in that niche. There is a niche for each one of the organisms by themselves. They vary in size. Those are the first 15. And then there's, in sort of descending abundance, all these other niches that we can quantify. A lot of these niches, as a log scale, have basically like one metabolite feature in them. We don't know what they are. We're going to ignore them for now. And we're just going to say, let's consider the first part of this matrix that accounts for about 80% of the total metabolites that we can measure. Okay? And so the idea is that this box right here is now our prediction for the competition landscape in our community. Right? There are certain organisms that compete a lot with other things, and there are certain that don't compete at all. And now the idea is, can we take this landscape and start to parameterize our consumer resource model? And we can do that pretty simply. We can actually use the abundance of the species grown in fresh media or in the spent media to parameterize our unknown resource levels. And then we can use the growth rates in these media to parameterize our rates. Right? We're just doing linear fitting, nothing complicated at all. And we get this. So now this is a predicted consumer resource model. It's a quantitative prediction for how any subset of these species should grow over time. Right? And up the top here is the prediction for the initial level of nutrients in each one of these coarse-grained resources. So the first thing we did was to try to predict how each species will grow. So the red is the prediction, and the blue is the experimental data. All the predictions are pretty good. This is not that impressive because we did use part of this data to fit the model in the first place. But, you know, it's a good starting point. Perhaps more challengingly, what Polly and Taylor did was to take random subsets of species, anywhere from 3 to 14 species, put them together, passage them to steady state, and ask how they assemble. Right? And so this is a random subset of the 200 different communities that they assembled. And you can see there that in general, it's doing pretty well in predicting how the species will grow to various abundances. In fact, the average error is about 1.3 doublings per species. It's about 2.5-fold, which is not perfect, but is actually pretty good even in comparison to just like sequencing error from batch to batch. Actually, one of the reviewers said, well, wait, you are not, you seem to be saying you can predict all the dynamics of this community. And so you should be able to tell me how every molecule in this community is being depleted. And they even said, that seems ridiculous. But we said, no, that isn't ridiculous. That is actually exactly what we're saying. So let's try that. So we took our 15 species, put them together. And then as they were growing up, we just took an aliquot every hour and did metabolomics on those samples. Then we took all the different features and grouped them together in the way that our competition landscape would predict, right? And so each one of these plots now is the dynamics of metabolite depletion of that coarse-grained grouping. And you can see here that it's not perfect. There's some cases like here and here where we get the time scale wrong. But on the whole, we predict the time scale of depletion. Here's the predicted depletion time scale. Here's the actual depletion time scale with a remarkably good correlation. And in fact, these 3 points here are particularly interesting because they are nutrients that end up being quite special, right? So they're sort of the exception that for us ended up proving the rule. So this was something we did in a rich medium called BHI. We also did the same thing in another rich medium, MGAM, where the nutrient competition landscape is totally different, but the strategy works exactly the same way and works with the same accuracy. We can actually then use the information from our model to make this prediction for in vitro growth even better. I didn't show you that here. But also we can even identify now those cases where you have like a positive interaction that's very clear. So this is actually the only case where a species, B theta, grew better in spent media from another species, shown here in the green dashed line, than it did in fresh media. And we know, actually, in this case, it's due to cross-feeding of hemin, which is an iron-related molecule. And we can actually explicitly incorporate this cross-feeding into our model and improve all the predictions that involve B theta and this other cross-feeding organism, EFERT. So I just say we were excited by this because it seemed, for us, it provides like a framework to start building more complexity into. Yeah. Yeah. Yeah, it's a good question. I mean, obviously, we can't do this with like 200 different communities. The reason we started with this particular community is we knew that the 15 species themselves recapitulate what we see in mice. We haven't yet done subsets because we're doing something slightly different to try to tackle this question. I think it's a very good one, and I think it will help to highlight where the in vitro, in vivo gap arises. But I can't give you a great answer because I think it's sort of a statistical question that requires sampling more than just like, a few communities. Yeah. So one of the things that also comes up, though, a lot when I present work like this is, why do you care so much about who is in the community? Right? Like, presumably, a large fraction of you have never heard of most of these organisms and could not care less which ones they are. And that's totally fine. And so I think what you do care about is whether your microbiota is doing anything useful for you. Right? And I at least care about that. And so I think we have started to think a lot about function in the microbiota. And this is actually a study that presented a perspective, I'll say, on what that could mean. So what I'm showing you here is from the Human Microbiome Project. Each one of these groups is different regions of the body for different human subjects labeled here. And then in each one of these groups, you can see the variation in microbiome composition on the top across individuals. Like, for instance, in stool, there's some people with a lot of the blue phylum and others that have a lot of the green phylum. On the bottom is that if you take these samples, you sequence their metagenome, and then you group them into metabolic categories, how do they group in terms of functional categories instead of species? And what you see here is that all of these are much less variable than these, right? And the idea that was put forth to explain this is that maybe microbiomes exhibit functional redundancy, that it doesn't matter who is there, but if one guy leaves and is doing one function, then someone else comes in and will do the same function, right? This has been observed in bromeliad communities as well. You can see here, this is at the species level and the family level, tons of variation between plants. But if you look at metabolic gene groups, you see much less variation, right? I just say, I'm sure there are many of you in the audience saying to yourself, what does it even mean, right? Like, yeah, like these lines are flatter than these lines, right? That is undeniable. But is that like even a relevant metric? Like, these are not, and you could think of these as not even having the same units. And that is a problem, but it's a very compelling picture. This is the phrase that has been used. It's the song, not the singer, that we should care about. And I think this has been a compelling picture. It's stimulated lots and lots of similar sort of measurements. And I thought it was a beautiful story. I just wanted to understand, like, what actually are we comparing against? And so what Poyi did was the following. He took the Human Microbiome Project data, and he generated in silico communities from the distributions of each taxon, right? So these simulated ecosystems here have no functional redundancy whatsoever. In fact, we're just going to define function by creating a random mapping matrix G, right? And then this is what happens when you create functional profiles, little g, from this random mapping by just transforming each one of these ecosystems, right? And lo and behold, these functional profiles, you can see here quantified on the right, are less variable than these taxonomic profiles. This is just the central limit there, right? We have many functions that are being averaged over many species. And so this is a statistical averaging effect. How can I show you that? Quantitatively, I can just scramble the species labels in this taxonomy function mapping and create a whole lot of random permutations, generate a whole lot of functional profiles. And sure enough, the data here for the average distance from the mean falls right in the middle of this null distribution because it itself is a realization of the null distribution, right? That makes sense? Okay, so this is basically just saying I can detect when I don't have functional redundancy. So now let's apply this to real data. So this is the bromeliad community. You can see here, this is exactly the same data I showed you before, just plotted in the same color scheme. Again, you see less variability at the functional level. Here is the mapping profile. And actually now what you can see here is that the data is even more variable from a functional perspective than random scramblings, right? So a sign of so-called functional redundancy would be if the red line was to the left of this distribution, right? I don't have any explanation for what's going on here, but I think we can say it's definitely not the sort of functional redundancy that you might expect. So does functional redundancy ever happen? Well, this is a beautiful system where we kind of knew it had to be there. So these were communities much like the gut communities I showed you before from Alvaro Sanchez's lab, where they took leaves and soil and they passaged them in vitro. And what you can see here is the taxonomic profile here. And then what they inferred was that, what they saw was that many different technical replicates from the same leaf would generate things that looked very similar at the family level, but very different at the species level. And so what they guessed was that maybe families have functional redundancy. And so what they used as their mapping matrix was the just the family association, which is shown here. And so what you can see here is that indeed the data here, the variability in the functional profiles is much less than anything you could get from scrambling. And this makes total sense because what we know about this community is that it's composed of one family, the blue family, that eats glucose, specializes in glucose, makes short-chain fatty acids, and these acids are consumed by the purple species. And so all you need is some blue guys and some purple guys, and you get a sort of equivalent level because they're interacting with each other. All right, but so you can see that in the data. So what about the gut communities that I showed you before? So these are the gut communities Po-Yi and Taylor work with. This is their family level profiling as determined by this matrix. And what you can see here is that it's a very different story in the gut communities, where you can see that now the data is actually to the right of the null distribution. But this actually shouldn't have been surprising, because I actually showed you at the beginning that these 15 species grow very differently, and there was no sense that species in the same family grew similarly, right? So what if we group them based on who eats similarly, right? Then this is what you get here, right? And so this is a very different mapping. And what you can see here now is that the actual data is to the far left of the distribution. It actually isn't completely outside of the distribution, which is interesting to think about. And so it might imply that there are even more signatures that we should be including in the mapping to make sense of true functional redundancy. But I think it highlights the fact that functional redundancy, of course, depends on how you define function. And I think if you're thinking to yourself, well, I believe the first part of your talk, and so aren't you just saying like exactly the same thing again? That is kind of true. But I think it also provides like a different quantitative way to ask, like, how well have we really understood this competition landscape? So let me turn everything on its head again to say that we went back to the Human Microbiome Project data, and we realized that A, we could look at it even more deeply, but also we thought maybe there is functional redundancy in the human gut. So here is, again, the profiles of different communities. And now we're just looking at one specific protein family, the glucokinase family, right? And we realized we can actually ask for functional redundancy on a family-by-family basis, right? And so, again, there's not much variability in this glucokinase family compared to what's going on at the taxonomic level, right? And what we noticed was that if we do the random shufflings, that the variability in the glucokinase family was actually lower than you would expect. However, there was something else we noticed as well, which was that if you just look at whether species do or do not have a glucokinase family member in their genome, that is actually correlated with their mean abundance. The ones that have this family are at higher abundance. And that's how you should think about any sort of core function in the genome, right? That is a selection, but it's a selection on each species. It's not an ecosystem level selection, right? For instance, ribosomes are just good. Don't have ribosomes, you're in bad shape. And that's regardless of who else is around you. And so what we did was we went back to our simulated ecosystems and we said, OK, let's now add to this one family of proteins, one function, where we're going to create a bias, where if you have that function, you will be at higher abundance, right? And now let's do the same thing. Let's calculate the scrambled matrices. And sure enough, the data is less variable than the null, right? So this is, again, a system where there is no functional redundancy. There's no selection on the communities. But we're seeing something that looks like that selection, right? And the reason is because in the random scramblings that I'm showing you here in the gray distribution, we are also completely changing the abundance bias, right? We've scrambled all the species. So it is no longer the case that this protein is associated with species at higher abundance, right? So we can fix that. What we can do is to, instead of just randomly scrambling all the species, we can scramble them in a restricted way and preserve this abundance bias, right? And the idea is we're seeing this reduction in variability. I told you at the beginning it can be due to statistical averaging. There may be this abundance bias as well. And what we're going to do is say, if we can get rid of both of those, maybe what we're left with is the functional redundancy, right? It's certainly, functional redundancy is not either one of these. These are not selections on ecosystems. And so one of the issues, well, sorry, so I should say, if you do this restricted shuffle, now what you can see here is that the abundance bias is preserved. It had to. That's how we were choosing the shuffles. But now this change goes away. So the gray is the normal shuffles. Now all the restricted shuffles have the same variability as the data itself, as it had to, because there was nothing else in the system. This is actually just a demonstration that we've taken everything out of the system that we put in, right? What's interesting is that I'm not going to get into in detail, but you can kind of think of these functional biases as being a product of scaling laws that seem to be implicit in a lot of microbial ecosystems, like scaling between the variance of a species with its mean abundance. But I won't get into that here. So let's go back to glucokinase. I mentioned before that it looks like there's some sign of functional redundancy because the data is to the left of the null shuffles. But if you do the restricted shuffles, that preserves the abundance bias. And now, sure enough, it's no longer less variable than you would expect, right? So this is not a functionally redundant protein category. Let's look at all of the 5,800 KEGG ortholog classes that the Human Microbiome Project looked at. We can look at the CV of the z-score as a metric for this difference in variability. You can see that for the random shuffles in gray. There's lots of things here that look functionally redundant. But if you take away this abundance bias, you go to the gray distribution, and it turns out that of the 1,800 families that might have shown some sign of functional redundancy, exactly 0 of them are statistically significant when you take away abundance bias. I don't know what that means. I find it interesting. I do think, in retrospect, I don't understand why anyone expected the human microbiome to show functional redundancy. Like, we all eat different things, we exercise differently, we live in different places. It's not clear to me why looking across a set of people would generate something that would preserve this sort of ecosystem level selection. On the other hand, that's not a proof that it didn't happen. It's just that with these null models, we can at least start to separate out using the data that we have. So some of you might be thinking, well, why are you talking about genomes anyway? Why aren't we looking at transcriptomic data, proteomic data? And if we do the same thing, we might find functional redundancy there. We just don't have that data, right? That wasn't part of the HMP data set. And what I would argue, at least, is that you have to deal with these sorts of null models to be able to even say something meaningful about those data sets. So I think the thing I really want to emphasize is this statistical averaging, because it's sort of undeniable across ecosystems like this. And frequently, it's basically just like a trick of the eye. You know, you look at it and you're like, that looks so much more controlled, but it's just, yeah, it's just an artifact. So in the last part, I want to think about what's actually going on inside a host. Yeah. I think it's an interesting idea. And I think all these things should be pursued. Metabolomes, proteomes, and transcriptomes. Yeah. Oh, sure. The question was, perhaps we should be looking at metabolites because, metabolites reflect kind of the currency, especially of interactions between host and microbes. I think it's a great idea. We just don't have the data to do that yet. So I mentioned this organism B theta. It's a fascinating model for human gut microbes. It has great fiber degrading capacity. And we were really interested in trying to understand how organisms like this change when they go inside a host. One of the kind of funny things is that I started having conversations with collaborators and saying, you keep putting bugs into mice, taking them out of mice. Are they the same? And they say, many of them would say, of course they're the same. And I'd say, well, why, of course? And then they would say, oh, good point. They're probably not the same. And I'd say, well, I mean, there's an answer here. We should get to it. And it turns out the answer is very rich. And so the answer the tactic we took was the following. We generated a large collection of barcoded B theta strains. So these barcodes are incorporated into the genome. And we created 2 plates of strains. Each well in each plate has 200 to 400 strains. So collectively, this is about 60,000 different strains. And we put these plates, we combine these plates, put some of them into one cohort of mice, the other plate into another cohort of mice. We actually also took the first cohort and changed their diet as well, took out all the fiber. And after 2 weeks, we took the plate 1 and the plate 2 mice, and we housed them together so we could look at transmission between them. So this enabled us to answer all kinds of questions that we were interested in. And I'm just going to take you through each one of them, hopefully in a simple way. So the first is, how fast is this organism evolving inside a host? Right? And we had no idea whether this was like days, weeks, months. Maybe it's so well suited to the gut that nothing is going to happen. And the answer is things are happening fast. So this is a Muller plot, which is showing you in colors, either blue or orange, the 250 most abundant barcodes. So they start out at very low levels because there's 30,000 barcodes in each mouse. So all the rest of the gray is the other barcodes. But what you can see here is that after 20 days, in all the cases, there's at least 50% of the community accounted for by just those 250 barcodes. Right? We can actually quantify the rate of evolution by looking at the Shannon diversity here. And you can see here that it's very reproducible across mice and across these 2 different plates, as it should be, because these 2 plates are just 2 basically biological replicates. If we change the diet, you can actually see that after 2 weeks, 50% of the community is a single barcode. And that's reflected in the faster dynamics of selection in these mice. So something about the diet can actually drive very, very fast selection. So remember, I told you that we're not just watching what happens in a mouse, we're also watching implicitly what's happening across mice. And having all these barcodes enables us to see those transmission events happening from very, very low levels. And so we took the 4 mice that have been co-housed. You can see here these 2 blue mice or these 2 orange mice. Different things are happening. You can see here that the blue barcodes come in right away after co-housing, whereas in the blue mice, the orange barcodes take a long time to come in. This presumably reflects a difference in fitness between the blue barcodes and the orange barcodes. And I should say, this hashed barcode, if I have time, I'll come back to in a few slides. So this reflects that there's asymmetry in the transmission between the 2. It's actually not just differences in fitness. We can quantify the transmission frequency directly by looking at the recipient versus donor mice. And you can see that it varies across mice and across time, which we think is interesting. But what we found really interesting is that each barcode seems to have its own signature of transmission. So we can define a transmission floor. This is the idea that all that's happening is that the mice are eating the poop of other mice. And so the barcodes come in, and then they just go straight out, right? And so that we can just look at based on the overall rate of transmission across mice. But then there's also an engraftment ceiling. So this is this is when there's actually a fitness advantage, and then that tells you how quickly that barcode will come in and reach the frequency that it was at in the donor mouse. So this is an example where the barcode actually engrafts even faster than you'd expect based on the engraftment rate across all barcodes. This is an example where it hits the engraftment ceiling and just stays there. This is an example where it stays near the transmission floor. This is an example where it stays near the transmission floor and then suddenly takes off. All of these different behaviors are happening, and they're all completely reproducible across mice, right? So every barcode will do the same thing as it goes into the different mice in the cage. So the question that was kind of motivating this whole study was actually this one, which is, how does the presence of a community affect what happens to the evolution of B theta? Everything I've been showing you before is just germ-free mice that have only this one species, this one barcoded library in it. Now what happens when we add the community? And so the idea was that there's two camps. There's the idea that because a lot of this, I forgot to say, a lot of the selection that we're seeing from a genomic level is on genes related to metabolism. And so if metabolism is what gets you a fitness benefit, then perhaps when B theta is by itself, it has a niche for tacos and hamburgers and sandwiches and chocolate. But now when you put in a community, it shuts down a lot of those niches. And all B theta can do is to learn to eat the chocolate. And so evolution is going to slow down. And then that niche preemption picture has an opposite counterpart, which is niche diversification. That maybe B theta can't eat the hamburger, but another organism is going to split up the hamburger into parts, and B theta can eat the tomato, right? And that would be niche diversification, where you would expect to see evolution speed up. So which one happens? Let's get a show of hands. How many people think the left? How many people think the right? Okay. I'll tell you the answer in a second. What we did was to take a synthetic community of 50 species, B theta plus 49 others, and then we looked at mono-colonized mice, co-colonized mice. We even looked at the effect of delaying colonization. It turns out not to have an effect at all. These 49 species all colonized the mice. We can detect at least 47 of them at most data points. And so this is what's happening in the molar plots, but just focus here on these Shannon diversity plots. I'm showing you the mono-colonized in blue and then the co-colonized mice in green. Turns out you're all right, because both are happening. How do I know that both are happening? It could be just that B theta is totally insulated from the community. Well, for anyone who's tried to make a library of 60,000 strains, probably one thing you know, these are bacteria. They're evolving all the time, right? There is pre-existing variation in every colony of bacteria. And it turns out that a small fraction of that pre-existing variation does confer fitness benefits. And so the reason we know there's a likely combination of niche preemption and diversification is because what we can do is to compare biological replicates of the experiment where we mono-colonized mice with the same diet with the library. And what you can see here is that there's a small subset of barcodes that have the same fitness in the two cases. That should not be true if they had no mutations to start with, right? So this is the pre-existent variance that we can then take to other conditions and ask, do you preserve the same fitness? And the answer is no. And so there is a different fitness landscape for a community colonized mouse or a mouse that has a diet change. If you'd like, what we're effectively measuring here is the distribution of joint entropy across these conditions. And so what we think is happening is that niches are getting shut down and niches are getting generated. So I think one of the things I'll just end with is to say that we can actually track transmission as well in these mice. And one of the things we noticed was that transmission can be wildly variable. So this is a case where they started out with the blue barcodes. The orange barcode set is actually only 100 barcodes, which is why it looks sort of amplified. And but what you can see here is that this blue mouse gets very little purple barcodes, but this purple mouse gets a ton of blue barcodes right away, right? So there's a huge asymmetry in the transmissibility. And what you can also see here is that there are also, in the community colonized case, there are some cases like this purple mouse, which got essentially no transmission whatsoever, right? So transmission can be incredibly low in a community colonized case, although that is not true across the board. You can see here in this case that this blue mouse got some of the orange right away and some purple later on, right? So there's no sort of like tried and true principle. I think this by itself is really important because when we interpret mouse experiments, we kind of like to think that the mice in the cage are having the same behaviors. And this is saying that at least at the level of what ends up in the gut, that is not true. I also want to emphasize that everything I've been telling you about transmission sort of goes against the stereotypical picture that we have this set of organisms in our gut and it maintains strong colonization resistance against other bugs coming in. These are bugs that are as closely related as you can imagine, and they're definitely transmitting and engrafting in the community. So I think there's a more nuanced picture of colonization resistance that has to emerge. I'll just end by saying this case here, where you have no transmission, I have no idea why that happens. I have a pet hypothesis that if anyone's interested, happy to chat with people about. This was in our gut biogeography paper, where we took a look at the gut and we stained the red species here that represent organisms like B theta. And what we found is that they're heavily organized on the outside of the fecal pellet. So this is the gut epithelium, and this would be like the edge of the fecal pellet. And I wonder if in the context of a community, the B theta actually gets pushed to the outside. And so now you're completely exposed to the outside environment, which has lots of oxygen when the poop comes out. And so they all just die. And that's why they're not transmitting, because that's that. But I think much more we'll need to do to figure that out. Anyway, what I'd like to highlight, and I think many of you who work with barcoding data have probably already drunk the Kool-Aid, is that the combination of barcoding and metagenomic sequencing data are a really powerful tool for starting to tease apart the evolutionary dynamics in a complex system. And much like the sort of resource competition I told you about at the beginning, it turns out these evolutionary dynamics are not as complicated as we thought. There's definitely behaviors we didn't expect to see, but they're highly reproducible. We can see the same things happening in terms of transmission across mice. And so sort of giving me a bit of hope that there's a sort of level of comprehensibility that we can attain in the future. So I think with that, I just want to highlight Po-Yi and Taylor, and especially Ben Good and his student Daniel, who are driving the evolution project. And thanks for your attention.